Hello, I am Rob David from Thermo Fisher Scientific, and this is Double Helix Diaries. In this episode, meet Steve Berosik, a molecular biology senior manager at Thermo Fisher Scientific. Today, we're going to ask him about the legacy and evolution of capillary electrophoresis. Well, Steve, thanks for joining us today on Double Helix Diaries. We are so excited to hear from you and your journey today.
Well, thanks for having me, Rob. I'm honored to be on the show.
Awesome. So to start us off today, we'd like for you to explain to us, if you could, a little bit about capillary electrophoresis technology, and specifically, many of our viewers associate it with Sanger sequencing. But certainly, as I know, and you from your experience, it entails a much broader scope than just Sanger. If you could elaborate for our audience a bit.
Yeah, our capillary electrophoresis instruments perform the process of separating DNA, dye-labeled DNA, by size and charge. This is done either through a sequencing reaction where you design primers for a targeted gene segment that you're going to sequence, and you sequence to the single base resolution, so you know exact base code of that DNA that you're sequencing. Or they'll design primers that dye-label a fragment of DNA that gets sized in fragment analysis applications. The process of capillary electrophoresis is automated on our genetic analyzers. The user will put one of these sequencing or fragment analysis plates on an instrument, link a plate record. From then, they'll hit start, and the process is automated. The capillaries will get filled with a sieving matrix or a polymer. Once the polymer is regenerated within the capillaries, the autosampler within these instruments will bring the plate to the electrode, and it will perform an electrokinetic injection. And an electrokinetic injection just entails a small low voltage applied to a sample for a few seconds to get a slug of sample injected into the capillary. Different than conventional thinking, folks think that these are manually aspirated by a pump, but we actually use voltage to get DNA sample into the capillaries. Once the DNA is into the capillaries, the autosampler will bring a buffer to the electrodes, which supports the electrophoresis once we apply a voltage. That voltage gets applied, and the DNA begins to migrate and separate by size and charge. So the smaller fragments will migrate quicker than the larger fragments. And as these fragments get near a detection area, it's called a detection zone, the fragments will get excited, the dye will get excited by a laser that hits the fragment, and that laser will emit a photon that gets captured by a CCD camera, and from that camera, data is processed into either size called data for fragment analysis samples or base called data for sequencing samples.
Excellent. Thank you very much for that. You dove right into the technical details of it, and I know a lot of our audience probably has seen the machines sitting on the benchtop, but they haven't necessarily understand how they work. So thank you for that. If we could maybe describe the applications that one would have for sequencing versus fragment.
Certainly, at a high level, sequencing with our capillary electrophoresis instruments entails a targeted gene sequencing. So you may target, say, a P53 or KRAS gene and want to interrogate for variants. So you'll amplify a segment of that DNA, sequence the PCR product, and interrogate for a position that you expect to see a variant in. Plasmid sequencing is also an application supported by sequencing where you can take a plasmid that you've grown up and purified, and you want to identify the sequence or confirm the sequence of the plasmid. There's also next generation sequencing confirmation where next generation sequencing might discover a bunch of variants, and CE application would then serve the purpose of confirming those because of the high accuracy of the capillary electrophoresis genetic analyzers that we produce. Fragment analysis, on the other hand, is a STR-based or standard tandem repeat type of an application where you are looking to amplify a portion of a gene or a loci and look for expansions or contractions within these repeats, and you size the various samples according to the number of repeats within the loci. Some of the applications that sort of technology would support might be our RHID application used in the forensic field. There are also copy number variation as well as SDR analysis like HID, cystic fibrosis, hereditary diseases, and pathogen detection types of applications supported by our fragment analysis type of work.
So if we could, so now we'll pivot back to the bread and butter application, of the technology Sanger, and specifically, we know that it was used quite a bit. In fact, it was the backbone of the Human Genome Project back in the early 2000s. Could you please shed a bit more light on what the various milestones during the development of the applications of the CE technology?
Certainly, yeah. I came on board Applied Biosystems in the early 90s in Foster City, doing protein sequencing on the 490 synthesizers and DNA synthesis on the 3948 DNA synthesizers. Early in the 1990s, I transitioned over to the CE R&D group, and we developed an automated, one of the first, the 310 actually is a single capillary automated genetic analyzer based upon the capillary electrophoresis technology. But during the mid to late 90s, we developed an instrument called the 3700. And what folks don't know about the Human Genome Project is initial discussions about how to proceed with that program started in the mid 80s, 1985 or so. It wasn't until about 1990 when the work got underway to actually sequence a human genome. And that work kind of trickled along. It was done on the slab gel type of technology that was very tedious. You had to pour gels.
That was the 377,
The
right?
377, yeah, that's correct. So it was a tedious manual process. Resolution was okay, got the job done. The read links weren't long, but the run times were somewhat longer than our current technology. In the late 90s, we emerged with an automated high throughput capillary electrophoresis, this 3700, that really pushed that program to the finish line. So within the late 90s, the last 3 years or so, 97 to 2000, there was an initial draft of the human genome announced in 2000, and I think the final draft was announced in 2003. After that, the predecessor, the instrument that came after the 3700 was the 3730, and then there were a number of genomes that began to be sequenced after the human genome was announced, such as the chimpanzee. The chicken came first. There was a chimpanzee in 2004, and then the corn genome in 2009. So within about 4 or 5 years, all these genomes began to become sequenced because of the automated technology that we produced. And from that information, we began, or scientists, the folks that use our technology began HapMap type projects where they were able to compare genetic differences between species and that. So really the emergence of the 3700 and subsequent automated capillary electrophoresis after that really enabled a lot of DNA to be explored and compared.
Oh, wow. I had forgotten all of the different benchmark genomes that had come after. Quite often we think, oh, we might have started with, another organism, but I know one, we needed the research into the human, but then, expanding it, and you mentioned the corn into plant research.
Yeah, the gut microbiome was also started. The research into that area was also started in the late 2000s as well. And that work continues to this day as well.
Always. There's one thing that's true. There's always more bugs. All right. So now we'll address what I would guess the one could consider an sister technology, or an evolution of the sequencing technology, and that's next generation sequencing. So it's, I would say, currently, and actually has gained popularity, widely used, of course. And it's important to acknowledge that capillary electrophoresis is irreplaceable technology, considering the sequencing spectrum, and I guess we'd like you to elaborate a bit more, provide some recommendations on when it would be the most appropriate to utilize CE, capillary electrophoresis, or next generation sequencing.
Sure, great question, Rob. I'll start with an analogy. Personal opinion. Um, Next gen sequencing to me is kind of like the ocean liner or the large vessel transporting a large load of materials from point A to point B. Whereas capillary electrophoresis genetic analyzers to me is like an Indy car or a Formula One car, which is much more agile, it's quicker to answer, that sort of thing. So, at a really high level, next gen sequencing is well suited for large genome projects, such as the human genome. If we had that in the 90s, would have been much quicker to sequence that genome. CE technology, capillary electrophoresis, is well suited for smaller projects such as small genomes. We sequenced COVID a few years ago, when that first hit the scene, but more targeted gene sequencing, variant analysis, where you want a quick turnaround on an answer. The sample prep for CE is much more straightforward and quicker. The instrument prep and turnaround time on the results that you receive from a capillary electrophoresis is also quicker. So if you want a quick turnaround on a result, CE is your best bet. And projects from say 1,000 to 1,600 samples seem to be well suited for CE. If you have a few samples to run, the beauty of CE is that we've designed our instruments to be scalable. So we've got four capillary instruments, 8, 16, 24, 48, and 96 capillary instruments. So if you've got a few samples to run, we've got an instrument that will fill that need. If you've got higher throughput projects that require more samples to be run continuously, then the 3730, 96 capillary instrument will support that need. But they're both technologies definitely have a place in the world here. Next gen, large genome projects. CE continues to be relevant with confirmation of next generation variant detection discoveries, as well as the targeted gene sequencing type of application. So I don't see CE going away anytime soon because of its scalability, ease of use, high accuracy, and quick turnaround time to answer that people really need at times. In addition, fragment analysis is well suited for capillary electrophoresis as well. If you have limited amount of sample, next generation sequencing often needs more sample to be run. So if your sample amount is precious and minimal, it makes more sense to run that on capillary electrophoresis as well. So fragment apps are more suited in general to capillary electrophoresis than next gen sequencing.
Yeah, I think many, myself included, continue to refer to it as the gold standard.
Correct. Yeah, that's true.
So Steve, I had forgotten how great a storyteller you are, and I got to tell you, I heard a great story about a certain visitor that came on site, and you were doing your thing, giving the story of Sanger sequencing. Do you know the story I'm talking about?
I think I know the story you're talking about, Rob. Was in the heyday of the genome sequencing era, and we were on site at the Sanger Center in the UK installing a number of instruments. So we were just installing our 3700 base. We had a number of lab technicians around a chalkboard. This was in the day before dry erase boards,
I love it.
We had chalk and a chalkboard, and we were walking through the chemistry process with the Sanger sequencing and how that worked in detail. And 12 or 14 of the technicians were huddled around this board. And as we're walking through and completing the explanation of how Sanger sequencing works and in the cycle sequencing reaction, this older gentleman steps forward and introduces himself, compliments us first and saying, hey, that was a very nice explanation of the Sanger sequencing reaction. And by the way, puts his hand out for a handshake and says, I'm Fred Sanger.
Oh.
And we were blown away and had talked the whole evening about having taught Fred Sanger the Sanger sequencing technique to the group of folks at the Sanger Center in the UK. So it's experiences like that visiting customers, the folks I've had an opportunity to meet, and I haven't spent a ton of time with a lot of Nobel Prize winners, but have been able to talk briefly with 4 or 5 various Nobel Prize winners, and that includes Fred Sanger. So, my experience and time, nearly half my life, more than half my life spent towards developing CE technology, and the opportunities and the folks I've been able to run across, and the great work folks are performing on our instruments and equipment just continues to amaze me. So, really appreciate those sorts of opportunities, and, blew us away at the time.
Wow, that's truly special. Yeah, that's the wonderful thing about science is you just never know what doors it's going to open for us.
Yeah. Yeah, truly amazing.
So Steve, I'd just like to say thank you for joining us today. It's been a great conversation.
Well, thank you, Rob. I appreciate the time and asking me on.
You got it.